The theme of this session is living in a post-antibiotic world, challenges versus opportunities. And we have our distinguished guests here. I'll start by the names that I have in front of me. We have Professor Jean-Marie Pages, okay? Professor Pages, you worked at the research unit at Marseille University. You also were born in France on November 15th, 1951. 2 days before I was born. No, I'm just kidding. Okay, and so we look forward to hearing from you some of your research and some of the ideas. Correct? And so we will start with you, if that's okay. And then we will go to our two other distinguished guests. We're very happy to have them with us. So we will get started, and I will introduce them after we hear from you. S'il vous plaît.
So, thank you for this introduction, and thank you to BVA 2016 to give me the opportunity to present some recent concept about antibiotic activity. So, antibiotic activity, I think this is a mistake, because if you take into account this slide, it's clear that after the antibiotic area, we have now to face to the antibiotic unsusceptibility. Why? Because if you check the interval between the antibiotic prescription, the first clinical use, and the first description of resistance against these antibiotics, the interval strongly decreases time after time. So it's clear that now, even we are speaking about recent antibiotics, we have a resistance against these new molecules. Why? Because you have to take into account the bacterial revenge or the bacterial genus. We have several kinds of antibiotic mechanism of resistance. For instance, and briefly, we have a target modification, mutation of the targets, typical case, GRAs and mutation of GRAs or topoisomerase, which impair the quinolone activity. This is a very efficient mechanism, but specific. We have also some drug modification. Typical example, the beta-lactamase, which efficiently cleave the beta-lactams. Also very specific. And we have the modification of membrane permeability. This is a mechanical barrier which are able to block the penetration of all antibiotic classes. Not only antibiotics, but also biocides. So it's clear that now we must dissect the molecular basis of this mechanical barrier. Why? Because during several decades, our objective is to increase the antibiotic efficacy, and now we observe that the main key is the intracellular concentration, and the intracellular concentration of antibiotics is governed by membrane permeability. And the membrane permeability, we have two processes, the penetration, the influx. And for instance, for beta-lactams and fluoroquinolones, the penetration uses the porine pathway to pass through the outer membrane and reach the periplasmic space for beta-lactam or cytoplasmic space for fluoroquinolones. In contrast, inside the bacterial envelope, we have an opposite pathway, which is termed efflux pump, which are in charge to recognize and pump out of the cell the antibiotics. And it's clear that the balance between these two processes, influx and efflux, directly governs the intracellular concentration of your active drugs. And we have now the first concept, resident time concentration close to the target. This is very important because if you think about the toxicity of the drug, if you decrease the intracellular concentration, you increase the threshold for instance, determining the minimal inhibitory concentration. And this intracellular concentration is directly related to the compound structure. To the bacterial membrane, for instance, the porine, which is the normal hydrophilic pathway which assumes the penetration for beta-lactams, but also lipopolysaccharide and lipid. We have also the diffusion pathway. Porine are involved in a great majority. I mentioned beta-lactams and fluoroquinolones. So the regulation of porine's expression is a very efficient way for bacteria to decrease the penetration. The expression and activity of efflux pump. It's clear that we have in all biological membrane the expression of efflux, because this is a self-promoting detoxification pathway for all living cell. And in bacteria, due to the overuse and misuse of several antibiotics, now we have no naive bacteria. All bacteria express efflux pump. And finally, the cooperativity between transporter, which is very tightly regulated by several regulation operants. So we have to face to this global membrane permeability. What about this resident time concentration close to the target? This new concept, and we have the accumulation, and the accumulation is directly associated to the influx rate, due to the, for instance, you remember the compound parameters, the presence of porine and so on, and the efflux plus degradation. And if you try to mention some example concerning the role of the penetration rates, you have immediately to think about, for instance, beta-lactam, which is a major class of antibiotics, but also beta-lactamase inhibitor. I am sure that you know the combination between beta-lactam and beta-lactamase inhibitor in order to overcome the resistance due to the enzymatic production. And if you check the presence of porine, which is used as hydrophilic channel for beta-lactams to penetrate through the outer membrane, you observe that depending on the type of the porine which is produced by the cell, we have different susceptibility. And the susceptibility is here presented by the minimal inhibitory concentration. And we observe that, for instance, in red, when we have this kind of porine, we have a very low minimal inhibitory concentration, which reflects the high susceptibility of the strain that produce the red porine. And systematically, in all Enterobacteriaceae, we have this kind of balance between these two counts of porine. But now, if you examine some isolate which are collected in a patient during antibiotic therapy, you observe that day after day, the bacteria contaminating the patient is able to manipulate the presence of porine in the outer membrane. And you observe that the first isolate is susceptible, and you have the presence of the two porine. And for instance, after 1 week treatment, you observe that the strain become resistant, and you observe that the strain don't express OMP-F or OMP-C purine. And the correlation is the high MIC observed for carbapenem. And we have several types of expression of purine, step by step, which is several levels of permeation, and the bacteria control the expression of the purine and control the permeation rate of beta-lactams. But we have the same problem with inhibitor of beta-lactamase. For instance, if you are a patient and if you are colonized by a bacteria which overproduce beta-lactamase, immediately the physician decides to use a combination with beta-lactams plus beta-lactamase inhibitor. But if your bacteria is able to degrade the beta-lactams, this is the beta-lactamase activity, if you added in vitro some inhibitor, you decrease the activity of beta-lactamase. But what happened in the cell? So we have a cell which produce purine, which overproduce beta-lactamase, and you observe that in the case of ceftazidime, which is a very current beta-lactam, in the presence of inhibitor, which is AVI, we have a decrease of minimal inhibitory concentration. So you win. But if your strain is devoid of purine, we have a high level of beta-lactam resistance, and you cannot circumvent this resistance by adding inhibitor. If you use a permeabilizer, you may restore a susceptibility. But this kind of membrane permeabilizer is not in clinical use. And we have the same thing with Enterobacter aerogenes, with Klebsiella pneumoniae, and a lot of Enterobacteriaceae. So it's clear that when you think about some problem of permeability, you have to integrate that you have to think about beta-lactams, but also inhibitor of beta-lactamase. So this is the first concept, resident time concentration close to the target. What about the kinetics of penetration and the kinetic of accumulation inside the cell? In order to investigate this various steps, we have developed several approaches in the lab, and we use, for instance, incubation with fluoroquinolones. This is very interesting because this is a fluorescent antibiotics. We make the incubation, washing, pellet, and you have two ways in order to dissect the intracellular accumulation. The first one is imaging by using microspectrometry, and you precisely localize the antibiotics inside the bacterial cell. The second one, by using the bacterial population, you dose directly the amount of antibiotic which have been penetrate the cell. And we observe, in this case, this is in normal condition, low-level accumulation of antibiotic accumulation in the cell that overproduce efflux pump. If you added in your incubation tube some poison which collapse the energy force necessary for stimulate the efflux pump, you observe a nice increase of the intracellular accumulation of your antibiotics. But what happened at the cellular level? You observe here, this is a micrograph of a suspension, and this is UV fluorescence imaging. You have the control cell without antibiotics, cell in the presence of antibiotics, no accumulation, and the cell in the presence of antibiotics and the presence of poison. And you observe that you have a nice fluorescence signal in the cell. But be careful with this final slide, you may observe that some cells are not fluorescent. And this represents the heterogeneity of the cell culture and the reality, because in your infection site, we have several growth levels of bacterial cell. And depending on the growth level, we have several responses about the accumulation. And this is the beginning of the pioneer resistance level, because if one cell escapes to the antibiotic treatment, step by step, you recover a bacterial infection due to this pioneer resistant cell. The second point is to investigate the kinetics inside individual cell. So you make the same experiments with an additional parameters. So you pick up the cell into cover slip and cover slip, and you investigate the kinetic of fluorescent signal during incubation with external antibiotics. And after mathematical treatment, you observe that during the time of incubation, we have a strong increase of the antibiotics inside the cell under the condition where you block the energy driving force for the efflux. And by using this kind of experiment, we are now able to have the kinetics parameters depending of, for instance, the expression of this kind of efflux pump. And by using this, you may obtain some clear kinetics idea about the accumulation. And you see, so the solid line corresponds to the efflux producer. In the efflux producer, we have a rapid steady state, about 5-minute accumulation. In the derivative strain, for which we have constructed knockout of the efflux, we have a continuous increase, and systematically, if you compare, for instance, the accumulation of the product A, this is a fluorofloxacin, and the other recent product produced by, for instance, GSK or AstraZeneca, we have the same type of response. And by using this comparative analysis, you are able to define a new concept about, the structure and intracellular accumulation relationship. By analogy to enzymatic approach, it's very important to define this SRR concept for each compound in order to have a clear view concerning the accumulation rate of this compound inside the cell. So, just to speak about the regulation of the membrane permeability. This is a sophisticated mechanism because the cell have a global response against chemical attack from the environment. The cell, the bacterial cell, is able to detect some aggressive compound in the medium. By using various, for instance, sensors, they are able to detect the concentration and rapidly to engage some response about impermeability. Just an example. If you incubate a cell in the presence of beta-lactams or antibiotics, 1 hour after the incubation, the bacterial cell decreases the expression of purines 1 hour after. So this is a very rapid event which are the origin of the rapid selection of impermeable cell. And we have, in addition, some global regulators which are able to manipulate not only the influx part, but also the efflux part. So, with the same regulator, for instance, RAM-A or MER-A regulation, we are able to decrease the penetration and to increase the efflux. And this is the reason for which, rapidly, the membrane permeability is the first line of defense for a bacteria. And this first line of defense is a very, very important thing for a bacteria. This is rapid, this is inducible and reversible, low cost, because during the absence of antibiotics, the bacterial cell is able to restore, for instance, the purine expression. This is a genetic and a biochemical regulation, because in addition, with this regulation of purine, the bacterial cell is able to synthesize specific compound, for instance, polyamine, and the polyamine is secreted in the medium, and the activity of polyamine is able to block the purine channel. So this is a biochemical resistance. This impacts various drug family. For instance, I have mentioned beta-lactams, quinolones, but we have also chloramphenicals, tetracycline, and so on. And this very efficient first defense line is able to favor acquisition of additional mechanisms, for instance, mutation in the target or expression of enzyme barrier. So this is very prevalent in all bacteria, and we have now to fuss about a phenotype which have been under-evaluated during the last decades. And we absolutely need a robust method to follow the kinetics and the accumulation level. This is a key point, because if you wish to project in the future, you have to design not only some new molecule which is perfectly adapted to the transport mechanism, but also to investigate the local concentration close to the target of your molecule. And I have just to acknowledge some intense collaboration which are very important during this investigation and the definition of this new concept with the Synchrotron Soleil in France, the Jacobs University in Bremen, University College of Dublin, University of Calgary, Fribourg, GSK, Sanofi, and a lot of members of my research unit. And also, you have also to mention several grants which is very important to develop this study. And remember, some old paper is also very important to prepare our revenge against the bacterial response. Thank you.
Can you hear me? It's very interesting. I think we have time for a couple of questions from the audience, if there is. Would you like, so that we can move on, because we have a long program here.
Thank you for this interesting talk. I'm asking about the mechanism of sensing the antibiotic in the media. Do we know the molecular biology of this mechanism, or can we target this mechanism in the bacteria? Is there any work done during this period about the mechanism of sensing the antibiotic in the media?
Yeah, at this moment, this is an interesting point, because if you remember, the expression of beta-lactamase is under the control of the AMP gene. And the AMP gene is directly controlled by some sensing mechanism. For instance, this is a two-component system which are able to detect the level of intermediate metabolite issue, for instance, in the case of beta-lactamase, to the accumulation of new piece of peptidoglycan. And if you accumulate new piece of peptidoglycan, nascent peptidoglycans, this is very dangerous, because this is intermediate metabolite and you may kill the cell. So the system detects the level of this intermediate metabolite and activates the beta-lactamase, which cleave the antibiotics, which is responsible for this accumulation of intermediate metabolites. And at this moment, we are investigating in my team some hypotheses concerning a similar two-component system which control not only the expression of beta-lactamase, but also the expression, the trigger of expression of efflux pump.
Okay. There's a question in the back, I believe. Please go ahead if you want to ask your question.
Thank you for your interesting lecture. I'd like to ask, I'm sorry if I missed it, but you mentioned that if you could target the organism with an antibiotic, including beta-lactamase inhibitor, it would be better. So if we are treating a case clinically, not doing cultural sensitivity, would it be better if we target both or adding more powerful antibiotics, including beta-lactamase inhibitor, or just start with the antibiotic without beta-lactamase inhibitor, like the first generations or penicillin? What would you suggest it better to start with? Thank you.
So I think this is naive and difficult question. So the first, my feeling is we have first to define the resistance mechanism which are active in your isolate. And theoretically, at this moment, currently, you have some probabilistic antibiotic, and if you identify the microorganism, you have some table which suggests you to use, for instance, ceftazidime or cephalosporin or quinolones and so on. And this is in blind assay. As a clinician, you have to treat the patient. So you treat the patient, but in same times, you have to isolate the pathogen and determine the resistance mechanisms which are active in the pathogen and adapt as soon as possible your treatment. Because, for instance, just an example. Several years ago, in Marseille, we carried out epidemiological assay in order to determine the membrane permeability involvement in resistance, for instance, in Enterobacter. And we observed that 10% of the clinical isolate have no porine in the membrane. So it's clear that for this 10% of isolate, you cannot use beta-lactams, and you cannot use beta-lactamase inhibitor. So it's clear that in same times you apply a treatment, you have to characterize the isolate. But remember, genomic characterization is not phenotypic characterization.
Okay. Thank you. I think we'll move to the next speaker. Thank you very much. Professor Martha Clockie is going to give us an interesting talk about, again, has to do with infection and antibiotics and you know, the interaction between some of these entities and how the information gained can help ameliorate some of the adverse effects of some of these diseases or some of the drugs that are used to treat these diseases. So, s'il vous plaît.
Thank you. Well, thank you for the kind invitation to be here. It's a great pleasure to be here in Alexandria. And I'd like to really continue from the talk that the last speaker did and continue focusing on resistance to antibiotics, which is a major problem throughout the world. So we've overused antibiotics and we've misused antibiotics, and we're now needing to find new ways to treat infectious disease. And my talk will focus on these things. These are bacteriophages, which are viruses that specifically infect bacteria. This picture, actually, this is the prettiest collaboration I did last year. I worked with an artist called Amy Lax, and she created this really beautiful sculpture of a Clostridium bacteria being infected by bacteriophages. And you can see what's happened here. She did this based on some electron micrographs I took. And then you can see what she's depicted as one of these viruses has already got into the cell and is replicated and is bursting out with more viruses. So they can be used, if you find appropriate viruses to target bacteria that cause us a nuisance, we can exploit these in a therapeutic way. And really the purpose of my talk is to give you more background and information on the subject. So, what bacteria do we have issues with antibiotics? Well, many, most. So I've just listed here the different types of bacteria that make us sick. So a third of the world's population carry TB, for example, and that is becoming increasingly resistant to antibiotics. So bacteria that cause food poisoning, or bacteria we pick up whilst we're in hospital, or just unfortunate enough to come in contact with. So a group of bacteria that are defined at the bottom here, known as the escape pathogens, are currently the most problematic bacteria in hospitals. But that's a moving target. So both different types of bacteria will make us ill, and different strains within these different bacterial species. So if you're going to use a bacteriophage, as I said, they have quite specific relationships. So you need to find a bacteriophage that's specific to that species and the subspecies of the bacteria that are making us ill. So just to illustrate this problem, this is just one paper of many that's shown. This is with time, and this is E. coli, Enterobacter, and Klebsiella. You can see with time there's more and more bacteria that are resistant to antibiotics. And meanwhile, we have an issue with the fact that there are fewer drugs coming onto the market. And the main thrust of my talk is that are bacteriophages a new treasure trove, as you like, of antimicrobials. They've been really very under-researched compared with many other conventional antibiotics, but as they're the natural enemy of bacteria there, they have a lot of potential. Really, without them, this is the type of headline that we're seeing at the moment. This was in the UK press last year. This is our chief government medical advisor called Dame Sally Davis, who said that the superbugs really threaten to return us to the dark ages. So we'll wind up going to hospital for something trivial, or, well, not trivial, something routine, and whilst they'll become infected with a bacteria which we'll then not be able to treat. So bacteriophages are one of many things which are being discovered. So this was researched again, and really this area has been really invigorated by this driver of antibiotic resistance. So this was a editorial in Nature emphasizing that fact, and also in Newsweek this was a review about bacteriophages, and I really like this quote from Roger Highfield. It says, "There is much we can learn from phages, even though they are nothing more than a scrap of genetic code and a protein coat." So really, as they've been evolving with bacteria since forever, so they've got all sorts of ways of being able to subterfuge and target bacteria. So we can learn a lot from studying this. So where are bacteriophages? I'm telling you, all bacteria have these natural enemies and we can potentially use them. Well, wherever you find bacteria in large quantities, you'll find bacteriophages. And I'm illustrating that with this picture here, which is showing you, this is just seawater that's been stained with a DNA stain, cybergreen. And you can see all these large particles are bacteria, and these little tiny particles are viruses. So really so even if you go into your own human gut, or you go into the puddles on the road, on the way to where you're staying, if you could examine the bacteria, you will find viruses trying to infect them. So you might be thinking, well, why viruses? So it's not just me who's telling you that viruses are important. I was part of a panel last year that was funded by the Wellcome Trust and the Department of Health in England to look at alternatives, to ask the question, where are our alternatives to conventional antibiotics going to come from? And of the top 10 areas that are being considered, you can see bacteriophages and phage products feature as 3 of the 10. So we've got wild-type bacteriophages, which is just natural bacteriophages, which is probably the first port of call. Although in parallel, people are also developing products based on lysins. So these are enzymes that are encoded by bacteriophages. So if you can figure out how a bacteriophage is doing the killing, you can then use the enzymes that the bacteria are producing. And it's not just, so lysins allow the viruses to get through the peptidoglycan at the surface of the bacteria. But it's not just lysins that phages encode. They encode all sorts of enzymes to get through biofilms, to get through the capsule of capsulating bacteria. So there's a whole set of really useful enzymes encoded by phages that we can, where there's still a lot of fundamental research that needs to be done to try and figure out in many species what these enzymes are and how they work. So as a young researcher, there's a lot of mileage to be had in this area, I think. And then the next thing that we can do, having found viruses, is really then work out to see if they can be improved. Can you broaden the host range, for example, to facilitate an easier production? Or can you give them additional properties which will help treat an infection? So that's the 3 major options when it comes to using a bacteriophage therapeutically. So using phages isn't a new idea. They were actually first isolated 101 years ago by an English person, and then 99 years ago independently by a French Canadian. And they are used, a lot of the early research was done in Georgia, and they're still used routinely in clinical practice there. However, the way in which data is collected has meant that it's not really transferable to using in many places. And so therefore, people have had to go back to the drawing board and start clinical trials really almost from scratch. There are some happening at the moment. There's a big one happening in France and Germany, France, Switzerland and Belgium with Fagoburn. There's one currently, Amplify, a US-based company, are also doing a clinical trial at the moment. And there's a huge interest really in the fundamental research. People are, even in the 15 years or something, I've been working exclusively on phages. You can see the amount of phage groups that are being worked on, and the size of the phage community is growing and growing, and not just by academics, but also you can see the big pharmaceutical companies, AstraZeneca, for example, are starting to become interested in bacteriophages as a therapeutic. So they also have what's known as GRAS status, which is generally recognized as safe. So they are used. You've probably eaten bacteriophages, well, you've definitely eaten bacteriophages just in the general consumption of your food. But if you've been to the USA and you've eaten hot dogs, you may have had phages which were sprayed on them, or also listeria-phages have been approved in the EU to use on cheese to prevent the buildup of listeria. And they're also used routinely in certain agricultural practices to prevent either pests or rot. So they are being used at the moment, but not really in a medical context. But this is changing. There are some clinical trials starting, but we still have a lot of research to do. So what makes them good? Well, they're very specific. There's limited issues associated with them because our bodies are full of them, so we don't mount a strong immune response, certainly when they're used topically or orally. So in a way, I think their main benefit is the fact that nature has already done the screening. So when you're looking for new antimicrobials, if you can find a phage, the phage has already evolved to kill that bacteria, so we can use something where the work has been done. We just have to figure out how to then use them. So they're probably going to be an important part of personalized medicine. We heard a lot about personalized medicine in the session this morning, and phages are likely to be a part of that. Well, if they're so good, why aren't we using them already? And really, this specificity that they have is also a disadvantage. So you really need to know a lot more about what you're trying to kill than you do with a standard conventional antibiotic. And because it was thought that they weren't really necessary after we discovered antibiotics, there really has been most phage research over the last 50 years has not focused on using phages therapeutically. So there's very little data on, for example, their dynamics and how you would use them, the dosage and the formulation. So this is what I'm going to, in the last sort of section of my talk, I want to tell you about some of the progress I've made in some of these areas in my lab and give you examples of this. So first of all, regardless of whether you're going to use a phage or a phage product or an engineered phage, you've got to find the right phage in the first place. So how on earth do you pull out that phage that might be a useful therapeutic? Well, I will take you on a little journey as to how you do this. So you need to understand where your bacteria is in a natural environment to hunt for those phages, which are the sort of winning phages in that setting. So here's an example of my two special little helpers that help me a lot with my phage sampling. This is sampling for Pseudomonas phages. They abound in muck heaps. And this is Clostridium. A lot of my work's been on Clostridium difficile. And I spent ages trying to find phages in guts of people that had Clostridium difficile. And actually, it's not a very good place to find them because when someone has an infection of C. diff, that bacteria is not associated with phages. But you also find C. difficile in the environment, in anoxic sediments and muds and rivers. And if you look in these environments, you find a lot more phages. Oh yeah, that's just a slightly puzzled expression of why he's being forced to sample muck heaps. These are some of my students. We're looking at viruses that target cyanobacteria in Africa. So obviously we're looking in these lakes to sample the viruses. And then I have a project where I'm trying to find phages, or I'm working on the phages that infect Borrelia, which is the causative agent of Lyme disease. So that's associated with ticks. So therefore, this is several of my students out last summer sampling in a deer park to get the ticks, which were then sampled for both the Borrelia and the phages. So you need to go to the environment when your bacteria are. Then, having done that, you need to enrich. So here we can see you take your sample, you enrich it in a culture of bacteria. You then filter the results of that enrichment, and then you can look to actually isolate bacteriophages. So you can see, to do that, you then grow up your bacteria, add your enriched sample, and if you're lucky, can you see here all these are bacteriophages now that it's each hole here on that lawn represents where a bacteriophage has gone in, it's killed the bacteria, it's released 10 to the 4, 10 to the 5, so it's released about 100 bacteriophages until you can see that plaque, so there's about 10 to the 4 phages in each little bacterial plaque. So then you then pick out those plaques, you repeat this process several times to make your phage clonal. And then you have the starting point of your treasure, so you can then see what the most valuable treasure is. So you can look to see what the host ranges of your bacteriophages. You can look at the genomes to check that they haven't got anything deleterious if you're going to use it in a therapeutic setting. So this is the genomes of various phages that we've examined. And then also you can look at the transcriptome. So we do RNA-seq work to see during expression what genes are up and down regulated to understand more about the mechanisms of these host interactions. We can look at the proteins to figure out what proteins, for example, are forming the structure of the virus. Because often when you sequence the genomes of these viruses, we're still at the point where a lot of the diversity is new. So perhaps up to half of all of the genes that you identify, maybe even more, have no recognized function. So we can also do metabolomics as well to try to understand more about these interactions. So really, if you want to use a phage as a therapeutic, the more you can actually understand about the way it interacts with the bacteria, the better. So, as I said, I've mainly focused on this guy here. This is Clostridium difficile, although I'll give you a little bit of some examples on Pseudomonas as well, because I've also worked on that a lot. And then we're working on strep pneumonia and, as I said, Lyme disease, and a little bit of work on other respiratory pathogens. We've got Borrelia here, Burkholderia here as well. So how would you actually use these bacteriophages? Well, you could use them instead of antibiotics. You could potentially also use them as a prophylactic. So if you knew somebody was going to come into contact with a pathogen, you could give them bacteriophages first before they even came into contact with it to prevent infection. You can also use them, particularly with Clostridium difficile. It's a disease where people have infectious diarrhea that relapses as soon as you take them on antibiotics, off the antibiotics. So you could give it to them to prevent those relapse patients. And a major driver really for using phages is where you do have antibiotic resistance. So again, that would be an area that would be useful to use them. And of course you can combine them with other therapeutics. So actually in our lab and other labs we've shown that if you actually combine bacteriophages with bacteria with antibiotics, you can improve the performance of the phages and extend the performance of the antibiotics. So that's how you would want to use them. So why Clostridium? Why am I going to give you a few examples of how we progressed the work in Clostridium? Well, that's actually the biggest cause of infectious diarrhea in the industrialized world. And as I said, it's found in many environments as well. But in hospitals, it kills tens of thousands of people worldwide every year. So it's very difficult to get rid of as well. When you get it into a hospital setting, it makes these spores. So these spores stick to the walls and float around in the air, and it's really hard to get rid of them. So the first thing that we had to do was, that first journey I took you on, we had to find the relevant phages. And this wasn't totally straightforward for Clostridium difficile. It's an obligate anaerobe, so you've got to grow it all anaerobically. However, we did manage to find, after a number of years, we've now found a lot of bacteriophages that target it, and they fall into three main groups. I've got pictures here so you can see what they look like. So there's three different types of what's known as a myovirus with these thick tails, and there's a different also Syphiviruses which have these long, non-contractile tails. And the reason why you need a good diversity of phages, if you want to use them therapeutically, is you want to prevent these problems with resistance that we've seen in antibiotics. You want to prevent the bacteria just becoming immediately resistant to the bacteriophages. And what I'm going to show you now is some data just on the ability of these different types of phages to infect Clostridium. And what I'm showing you here, this is a graph with phages along the bottom and strains along the top. So you can see that some of these phages really are not very useful at all, where you can just see a single square. A square just depicts a successful interaction. So where you can see a single square, that phage would be no good because it's hardly targeting anything. So you want to try and find phages with nice, broad host ranges. And these are my top 7, which I'm now working on to develop as a therapeutic. And all I really want you to see from this is that, well, to get the fact that I've isolated hundreds of phages, and then from that, based on what they target by looking at their host ranges, I found 7 phages that can actually infect most of the clinically relevant and prevalent species of Clostridium difficile. So having done that, we then look to see, well, can they actually kill in both in culture, in artificial systems, and then also in vivo settings. So before you go into clinical trials, you clearly need to have in vivo data to show that they'll actually work. And what's interesting is that if you this is just showing time, the ability of phages to kill with time, and laser pointer isn't really working, but what you can see is that, can you see the axis along the top of the graph? You can just see that those individual phages are not completely killing the Clostridium in a culture, whereas you can see some of these, the blue line, you can see that the Clostridium is being killed, but after 24 hours, it starts to recover. So what we've shown is, and other people have shown this in other systems, if you use one phage, you kill the bacteria, but resistant bacteria will recover and grow. Whereas if you use mixtures of phages, so that nice diversity of phages I showed you, you can completely prevent this, and you can get rid of every Clostridium cell in culture conditions. And the reason for this is that you can see here is that this is now showing you the Clostridium cells down here and phages along the top. And if you see, essentially, you start off with a wild type host which is sensitive to bacteriophages. Now, you can make host-resistant, phage-resistant mutants of that strain. So if you just look, can you see there's a 0 in the second column down? That means that when a phage-resistant mutant of the original strain cannot be infected by that phage, but they can be, and you can see this throughout the table, they can be infected by the other phages. So I showed you those 3 different types of myovirus and one type of Syphivirus. So when the cells become resistant to one type of virus, they're not resistant to others. So that's the theory behind why, at the moment, with natural phages, you'd use a mixture of these different types of viruses to prevent infection and resistance buildup. So what about model systems? Well, ideally, we need to show that they work in animals and in humans. So I've done quite a lot of work in a hamster model, mainly for Clostridium, because the mouse model, mice are not sensitive to the toxins that Clostridium makes, but so the hamsters generally are used as a good model for infection for that organism. But then we've also been developing other methods to avoid having to use too many animals to do this work. We're looking to see if studying the bacteria in cell lines and in insect models, what data we can get from that. And that looks rather effective. So this is showing you now in cell line models. And you can see that with time, this is where you can see it rather nicely at the bottom here. The eukaryotic cells are shown in the human epithelial, gut epithelial cells, shown in blue. And you can see the bacteria that are stained in red. And then when we've added bacteriophages to these infected human bacterial cells, we can see that the phages work well in that model, in the epithelial model. We've also got this insect model working well. This is a nice model. As you can see, it's with these little wax moths larvae. So when they're active, you can see they're wiggling away and they're white. And if they're as soon as they become sick, they just die. They turn black. So you have a good output to see. And we've got this model working really well for Clostridium and Pseudomonas, and we can see that so we can then start to test things, as I say, like the dosage of the phages and which combinations are going to work best to kill in a more realistic infection setting than just in a flask. So what about animal data? Well, this is the animal model that we've used, as I say, the hamster. And what we do here is that we take the hamster and we treat it with clindamycin to get rid of the other bacteria that are inside the gut of the hamster and to induce the Clostridium to germinate. And we then treat every 8 hours with the bacteriophages. And for this experiment, it was carried on until 36 hours. And this is the data. You can see that when we've treated with these are the control plots here, and you can see that the amount of bacteria that can be recovered from the cecum of the hamsters in a control animal is about 10 to the 6 bacteria per mil of the associated tissue of the cecum. When we've added bacteriophages, we can reduce the amount of colonization by several logs. So this is comparable with studies of other novel antibiotics, and it looks very promising as an approach. The plot on the right-hand side shows now that you can also, in a different model with the hamster, we can double the life expectancy following the treatment with phages. So the data is looking really quite promising in this particular setting, and I've sort of taken you through the steps that we need to do to show efficacy of bacteriophages or phage products. Mm-hmm. Now, the last sort of data slide I want to show you is now looking at the delivery. Because obviously if you make a phage as a therapeutic and you want to use it, you need to fit in with standard treatments of infectious diseases. So with Clostridium, for example, we're now developing a pH-sensitive polymer capsule that you would give to patients. In the hamster study that I showed you, we just gave the hamsters bicarb to neutralize the stomach acid. But ideally, you'd like to encapsulate those phages so you can know that they'll survive the passage of the acidic stomach and then be released in the colon. With Pseudomonas, one of the main diseases that Pseudomonas is associated with is cystic fibrosis, and that's currently treated. Antibiotics are given to patients through nebulizers. So we've tested different phage combinations with different nebulizers, and we can see that different nebulizers are a lot more effective than others. But the good news is phages do survive that nebulization process. So you can see they can be nebulized to the correct lower bronchial areas of the lungs. And some nebulizers are better than others. So for example, in this top design of nebulizer, we could get about 15% of all the phages that were input made it to the right area. So just to wrap up, we know a fair amount about very, very few phages. So if we are going to use bacteriophages therapeutically, there's a huge number of species where we can really, out there that have had their phages very poorly characterized. Carolina made the point with her talk yesterday that we have issues if we can't grow bacterial hosts, or bacterial hosts are difficult to work with, and very often people haven't really done an awful lot of work on their associated with bacteriophages. So for example, that Berelia project that I'm doing with Lyme disease, it's a really important disease, and the phages just have not been touched. And there are many other diseases out there that cause a problem. There's lots of work to keep you busy for a long time. And hopefully I've really shown you that, well, probably you already knew that antibiotic resistance is of genuine concern, but hopefully I have shown you that phages are likely to be a part of the solution to target it. I've shown you that they work in different model systems. So for example, in Clostridium, we're now at a position where we can do more work on the dosage and timing, as well as looking at the practical constraints of how you actually make the product and will deliver it. And at the moment, we've also been developing in our lab these models to then test new phages as we pull them out of different natural environments. So it just remains for me now to thank my lab. This is a picture of my lab last summer. We have an annual trip to the Leicestershire Canal systems. This is various my students and postdocs enjoying a nice day out. And obviously, I would like to thank the funders that have facilitated this work.
We'd like to thank you
for your
very
attention.
much for a wonderful talk. This is truly fascinating and very exciting. Opens an awful lot of ideas to do things. So thank you so much for, wonderful talk. We have time for one question, one burning question.
There is one. Yeah, there is. We'll probably take somebody who didn't ask before, so because it's one question. Sorry.
Thank you, Martha, for the wonderful presentation and the insight of bacteriophage therapy. When I'm teaching this material to my students, I have always an urgent question that I keep receiving from them, that looking back at all the virus evolution and their contribution in the evolution of other organisms, how safe are we from bacteriophage therapy looking at the future? Like, I know all our human bodies, we are consuming phages on daily basis, but when you're introducing phages in high doses to control a disease in a human body, that will mean that in several years from now, maybe the virus community in our body will change, in our gut will change. So how safe is that in terms of changing of the microbiota of the gut and our health in general? Thank you.
Yeah, no, it's a good question that your students ask you. I think that there's a lot of experimentation. Well, I think there's an awful lot to be worked out, first of all, in terms of the dosage. So you wouldn't necessarily be using really high dosages, and you also wouldn't be using them as a long-term, in the long term. So in terms of the effect on the microbiome, I think probably phages are going to have a more, less of a hacksaw approach than many antibiotics that we're using, which is just getting rid of everything and then we're having to recolonize. But I still think there's probably is a lot of work to be done in this area. So looking at we're about to do some work where we just actually look at the viromes and the actual resultant bacteria following treatment with phage therapy, because there hasn't been an awful lot of data on that area. So I think that we can't, although the risks, I would say the risks, based on what we know, are probably not high that we would cause massive, or will cause less severe impacts than we will with antibiotics. I still think that there is work to be done in this area to actually see what those impacts are. And I think even things like host range, often we don't test host range on hundreds of things, right? We find phages that infect one thing and we don't know about every single commensal bacteria in the human body. But I think that the specificity is on our side in general.
Thank you so much again for a wonderful
Okay, so I'm only sitting here to introduce the next speaker, who is also the chair, Dr. Malak Qadb, Professor Professor Mala Kotb is now at the University of North Dakota. Well, the reason I'm presenting her, she was my PhD advisor. So Dr. Kotb was actually born in Egypt, and she graduated from Ain Shams University Faculty of Science, Kulliyat al-Ulum. She's very proud of her class. One of them was the Minister of Research, Dr. Nadia Zakhari. And then she got her PhD in Memphis, Tennessee, in the United States. She went to the US very early in a time it was really hard to be there right after the October War.
Okay.
And then she stayed there. She got a position, a postdoc position at Duke, and then she went back to Memphis, Tennessee, and then she chaired a department in Cincinnati and then in North Dakota. We're very pleased to hear from her another side of the story. So regardless of antibiotics therapy, what happens to the host? What is the role of the human host in responding to infection? Some people get sick and some don't. So please join me to greet Dr. Kotb, who is giving the next talk.
Thank you, Rami, for a very nice introduction. Thank you all for being here. This has been an amazing conference, a lot of new data and exciting interactions. So I'm honored to be part of it. So I really appreciate it. We're having a little bit of problems with the computers, but hopefully they will behave nicely. And I am going to share with you what we're doing currently, what we've been doing for the last couple of years. We've been collaborating with a very large number of investigators in Europe and in other countries, France, Europe, Germany, on a very exciting project called INFECT. And the INFECT stands for the necrotizing fasciitis, which is a very severe and morbid disease that happens after, when the skin is subjected to a pathogen or there is aberration in the skin. So the acronym for our project is INFECT, and it is funded by the European Commission, by the Swedish Medical Institute, and by several of our personal grants as well. So you can see, so it stands for improving outcome of necrotizing fasciitis and elucidation of complex host pathogen and signatures that dictate severity of tissue infection, because these infections are really very nasty and morbid. Okay, so we got together and we formed a consortium so that we can collaborate and take advantage of each other's unique expertise and knowledge. In this particular case, our lab had a tremendous knowledge in animal models and how to standardize the animal models for research, so that to minimize any ambiguity or errors or to miss something. So we were asked to be WP1, which is the main lab that produces the data, and then we parse the data that we generate to all the other partners. So the people that need to do the artificial tissue do it, the people that need to do specific intervention and drug development will do it. So shown here is the table of all the participants, and the red arrow indicates us at the University of North Dakota. And we, of course, we were very interested in the NSTI murine model of infection, and we took a forward genetics approach to understand the underlying mechanism of this particular disease, which is very morbid, by the way, and very deadly. It's not a simple disease to have. As I said, there we are partners and we are, each partner is assigned certain duties. We happen to be partner number 1, WP1, and the reason we are WP1 is, again, as I just said, because we have tremendous knowledge of handling mice and doing translational research with animal models. And the data that we generate, we share it with the individuals that are doing the NSTI patient care, and of course the individuals that are doing modeling and integration of data coming from the other partner, WP4. And of course, we have a lot of samples from patients, and we have tremendous, meticulous documentation of every single thing that happens to either the patient or to the mice that we are using, because we don't want to subject the mice to any harm or pain, but in the same time we want to get the information. For that reason, we actually have shifts ongoing all night, during the day and all night. And people come to the lab to make sure that the animals are okay, and if the animal is suffering, that animal is sacrificed. So we're proud that we are all agreed to make sure that we are handling those animal models in a humane manner, and I think this actually helped us gain much better and much more reliable data. So again, WP1 is us developing the models, and WP2 are the patients actually and clinical investigation of the disease and the underlying mechanism of the disease. And WP5 is really the group that looks at the patient tissues and trying to do construct or develop artificial tissues so that they can heal the horrible lesions that the patients with NSTI usually get. It's a very horrible, really, and morbid disease. And then there is the modelers that model the host-pathogen interaction and model the dynamic of the disease and all these interactions. This is WP4. And then we have the artificial tissues, again, group, which to make sure that they can generate artificial tissues, and these are WP6. And then we all disseminate the data amongst ourselves. We have either conference calls or we have frequent meetings to make sure that the data is shared and that it is entered in the proper way. This is extremely important. Otherwise, garbage in, garbage out. Okay? So let's see, next slide here. In the first couple of years, we were interested in looking at genes and quantitative trait loci. These are loci on the mouse chromosome that harbor genes that are involved in modulating the severity of the disease or protecting against the particular infection. So we were able to map several QTLs. These are quantitative trait loci. And we mapped very, very significant QTLs that deals with survival, modulates survival. Another one for weight loss, and a third one for lesion size, the lesion size of the infected mice, okay? So that was important to establish that. And when we had this information, we had another tool called WebQTL. WebQTL allows us to analyze the data, the genomic data, in the context of the phenotypic data, and to elucidate underlying mechanism of how this abnormality happened, or how this mice, for example, had more severe disease than the other. So this had to be standardized. For that, we used, as I said, this wonderful tool, WebQTL, and we did one set for survival, another set for weight loss, and another set for lesion size. You can see peaks. I don't know whether you can see them from here. Yes? The genes under those peaks Okay, we inspect them very carefully because they are involved in modulating disease susceptibility and severity. So by focusing and extracting those genes and understanding what they are, we can then extract the underlying mechanism by which the severe disease develops, and also we get ideas about how we can treat those morbid diseases. So there were three main phenotypes that we started with. After that, we actually went to other phenotypes as well. But we had survival, QTL, quantitative trait loci. We had one for weight loss and one for lesion size. Okay? And then when we started to parse them into pathways, the genes that we were discovering, so we started to parse them into different pathways. We were really surprised because this wasn't something that we had predicted, that IL-1 beta was actually the key cytokine that was orchestrating the whole outcomes of the disease and the severity of the disease. This wasn't something that we actually had thought about, but it did. And as you can see here, it kind of like is the pillar of the whole study there. And we were able to identify 4 QTLs that were associated with, and affected by IL-1 beta. There was actually a total of 516 genes that we looked at, and from there we got the 4 QTLs. We also were able to identify 375 genes contributing to the severity of necrotizing fasciitis and STI. And we rank ordered those genes, based on, to make a short list based on relevance and potential for contribution to disease. We validated everything that we did with using quantitative RT-PCR, and we documented everything very carefully, and we ended up with 125 genes that were significantly differentially expressed between the protected and the susceptible mice. And then when we parsed these 125 genes into their respective pathways, we got what this map of the gene network, and this network that is interacting with each other to modulate the severity of the disease to make it either more severe or to protect at times. And from the study, we found out, as I just said a few minutes ago, that IL-1 beta was really a major regulator and played a very important role in reducing the pathology of the disease. Okay, so that's really the part. So when we had this information, the next thing we wanted to do is to look at other molecules that also contribute to this particular disease. And we knew, or we had some data that we had generated in our lab, that variation in MHC class II molecules play an important role in modulating the disease. Why? Because MHC class II molecules present superantigens to the T cells. Superantigens are super antigens. They are very aggressive. They can cause a lot of pathology, and that's why they're called superantigens. And it just happened to be that we had worked with the superantigens for years before we even started on this study, so we knew a lot of things about it. So we were able to look and see what if the specific HLA types, HLA class II types, are associated with either protection or predisposition to the more severe form of the disease, and we were able to actually identify them, and then we manipulated the system to make sure that these are actually performing the functions that we had predicted. Okay, so that took a lot of times to standardize the system, understand the variables, challenge ourselves to make sure that we're on the right track. And then in year 3, we complemented our unbiased systems approach because we were going in without any preconceived as to what the data will be or what the results will be. But in year 3, we were able to complement this unbiased system genetics approach, which is a very powerful approach, by the way, because it tells you the truth about what's happening. And we used a reductionist approach that included the use of humanized transgenic mice, which were a part of the HLA transgenic mice. And we also generated new mice that expressed HLA molecules that were associated with either protection against severe necrotizing fasciitis or predisposing to severe and morbid necrotizing fasciitis. So we also generated those constructs and used them to check the system. So why focus on the HLA class 2 and modulating the outcomes? This is because in our clinical study, we had done a huge clinical study with our Canadian friends and collaborators, and found that variation in HLA molecules played a very important role in modulating the severity of these infections, whether it was the necrotizing fasciitis infection or even toxic shock syndrome. So we did a very large translational study. We recruited a lot of number of people. And if you would forgive me, let me just tell you a story that I love to tell, because this was one of the best parts of my life. As we were working on this and trying to design this study about looking at the effects of the HLA class 2 and so forth, what I decided to do, I said to everybody, I said, we're going to get away completely. We're going to go to a very nice place in Florida that I had already rented a mansion. It's in Seaside, Florida. We're going to take our food with us, our drinks, everything. We're going to lock ourselves and discuss how are we going to design the big clinical study, what are inclusion and exclusion criteria, what would be things that we really need to monitor on a regular basis and so forth. So everybody, we went together. It was wonderful. We had to drag people from the beach to come back and finish their work, but it was the best thing we did because we really got solid data from doing this and attending to the small things that you may overlook, but they actually contribute importantly to the disease process. So in summary of this part, we found that host factors, especially human leukocyte antigen factor, HLA molecules, class 2 in particular, were expressed on the antigen-presenting cells, and they played an important role in determining the severity and outcomes of gas infection. So that gave us a way to start at least thinking about some treatments to do that. Before we did that, we went and looked at the association of other molecules that could confer risk or protection against this necrotizing fasciitis. And indeed, we found some haplotypes associated with the different outcomes. Specifically, those haplotypes, the DR15 and the DQ6 haplotype. Haplotypes mean genes that are linked together in one stretch, and it makes a haplotype. So we found that the DR15 and the DQ6 were highly protective against severe necrotizing fasciitis, which was a fantastic thing to find because this way it gave us some ideas about how to treat patients. On the other hand, we found that individuals that had the DR14, DQ5 haplotype were really at a very high risk of developing severe disease, and also there was a lot of morbidity and death in this particular group. So this was for this severe disease and toxic shock syndrome. Also for necrotizing fasciitis, we discovered that individuals with DR3 and DQ2 were protected against severe disease, but only in the absence of multi-organ failure. If they had multi-organ failure, they were not protected. For the risk also, we found that the DR14 and DQ5 genes were really conferred risk, or increased the risk of severe disease, but only in the absence of severe systemic disease. We also were able to identify that severe systemic disease plus necrotizing fasciitis, and it, of course, had a very undesirable outcome. But the DR15 and the DQ6 were not really protective against necrotizing fasciitis, but that they protected the onset of severe systemic disease in patients that had necrotizing fasciitis. So, there were several levels, and we were able to really understand these levels. Why were we pleased with that? Because that gave us a roadmap to allow us to target things. Instead of trial and error, we knew the underlying mechanism by doing these studies, and that's how it went. And actually, I have to say that every single young faculty and student that worked on this study got an international and national awards for the whole study. The PIs were just happy to stay back and cheer them on, but it was a very well-designed study, I have to say. Okay, so one of the things that we also looked at the time, because it was important, are superantigens. As many of you may know, there are regular antigens and so forth, but superantigens are super in the sense that they elicit very powerful inflammatory immune responses, okay? And they do so by binding to the MHC class II molecules, and also they bind to the T cell receptor on the T cells, and they transmit signals into these two cells, and these signals are translated into very strong inflammatory responses. And then there's a differentiation into these different cell types, most of which are really cause a lot of pathology and requiring amputation of limbs sometimes and so forth. So superantigens cross the V beta chain of the T cell receptor that's expressed on the T cell and the HLA molecules, and that is expressed on the antigen-presenting cells. This induces the potent stimulatory signals and exchange of signals, and drives the differentiation of the immune cells into functional types. These are the different functional types here, some of which are protective, like the Tregs, for example, but most of others were really can cause pathology and amputation and death in many cases, unfortunately. Okay, so that was the summary table that we found, and we were able to find which haplotypes and which genes were associated with what.
We
start.
We have 2 minutes.
Am I done?
2 minutes.
2 minutes? Okay. Everything went well after that, and we went home and enjoyed ourselves and went back to Seaside, Florida. No, I mean, this I'm actually done. Okay, so we were able to really elucidate the underlying mechanism of the disease. We used this information to develop targeted intervention and prevention and to treat patients properly. And that was really a very good outcome, and NIH was very happy with that. Okay, I'm not going to go into details with this. This is just surface expression of DR and DQ alleles on certain areas and certain parts of the body of in this particular case, the mice. And what are they doing there, and what is the pathology that comes up from there, and how can we tell with flow cytometry and antibodies how to deal with those invasions. And then we also excised the lesions that the mice developed, and we studied them to see what kind of cells are coming into the lesions so that we can block that. I have to tell you again, because I think I already said that, we had people going to check on the mice all night long and all day long, because if they found that a mouse is really suffering, this mouse had to be sacrificed, okay? We did not want to have any suffering in the mice that we, and that made us feel better about it. So,
Can we,
in summary, these are the results.
can we just skip to
The
conclusion?
the mice developed different lesions. We quantified those lesions. There were two main isolates, the 5448 and the 2006, and we got the data on both of these isolates. One was very severe, the other one was milder. And I think that I should stop here and refer you to some of our publications. And the HLA types are all published in our work if you want to look at them. The big database is available for anybody to mine or see. If you have any questions on including on multiple organ failure, please call us. We're more than happy to help. We're happy to help you design the study, interpret the data, and so forth. Thank you, and sorry.
Thank you so much, Dr. Malak. I think we have, we still have a chance for one burning question and one non-burning question. If anybody has a question to Dr. Cott, please try to find the mic.
Did you raise your hand?
There is a question. So until the mic goes there, maybe an obvious question, Dr. Malak?
Mm-hmm.
Maybe an obvious question would be like, how is this relevant to humans if you just do it on the mice?
Oh, it translated beautifully to humans. You know, when we had humans infected with some of the same pathogens, and we looked at the, the mechanism, it translated beautifully to humans, so it enabled us to actually administer and we collaborated with clinicians. Instead of amputating patients, we were able, we meaning the team, the clinicians and the researchers were able to actually preempt that. And many of them recovered very nicely and went on to live with their limbs intact and so forth.
We'll have one more question from the audience. Please go ahead. Oh, it was the same question? Oh, I read your mind. So we can have another question if somebody has a question from the audience. All right, so...
okay, can I say something?
Yeah.
If there are no questions, let me just, to the young people and especially and the researchers, keep in mind, the most important thing to do is to really spend time organizing and designing your study. This is think about everything ahead of time, because then you will get good data. If you don't do that, it will be garbage in, garbage out, and you will not benefit the patients. So this is very important to remember, and that's it.
Thank you so much, Dr. Malak. So I guess there is another session that's going to start here. So I would like to just that you all join me to thank the speakers one more time for all the panel. Thank you.